Post-translational modifications in the secretory pathway. Welcome to today's lecture on cell biology and, specifically, the fascinating topic of post-translational modifications in the secretory pathway. In this lecture, we will dive deep into the intricate processes that occur within the Golgi apparatus and its importance in various cellular functions. Understanding these mechanisms will not only enhance your knowledge of cellular biology but also provide insights into potential therapeutic approaches for diseases associated with Golgi dysfunction. So, let's begin our exploration. Protein trafficking through the secretory pathway involves the movement of proteins from the ER to the Golgi apparatus, where they undergo PTMs. These modifications, such as glycosylation, phosphorylation, and proteolytic cleavage, impact protein structure, function, and localization. The Golgi apparatus acts as a key site for glycosylation, adding sugar molecules to proteins, leading to the formation of glycoproteins. These modifications influence protein stability, targeting, and receptor recognition. During retrograde transport, proteins and lipids are packaged into Copy vesicles, which transport them from the Golgi back to the ER. This process, mediated by coat proteins, helps maintain the integrity and composition of the Golgi network, ensuring accurate protein sorting and overall Golgi homeostasis. Overall, the Golgi apparatus has a specialized machinery for retrograde transport. The cadal receptor recognizes luminal proteins with cadal or related motifs and recruits the Copy coat complex for vesicle formation. Golgins aid in vesicle fusion with the ER membrane. These regulatory mechanisms ensure proper protein glycosylation, which is essential for normal cellular function. Dysregulation of Golgi protein glycosylation can contribute to various diseases, highlighting the importance of understanding these processes in medical research and clinical practice. Traditionally, the Golgi apparatus has been associated with protein secretion via the classical pathway, where proteins are delivered to the plasma membrane. However, emerging evidence suggests the involvement of the Golgi in non-canonical secretion mechanisms. Non-canonical secretion refers to the release of molecules that do not rely on the classic secretory pathway. This includes unconventional protein secretion, release of extracellular vesicles, exosomes, and shedding of membrane-bound vesicles. The Golgi apparatus is implicated in the biogenesis and sorting of these non-canonical secretory vesicles, which serve as mediators of intercellular communication and contribute to various physiological and pathological processes. Rab-GT possase, small proteins, regulate Golgi to cell surface transport. Rab1 is involved in ER to Golgi transport. Rab6 is involved in Golgi to cell surface trafficking. Rab-GT possase recruit effector proteins to facilitate vesicle tethering, membrane fusion, and cargo recognition for accurate and efficient transport. In summary, the Golgi apparatus is essential for maintaining cellular asymmetry and specialized functions in polarized cells like epithelial cells and neurons. It facilitates the sorting and delivery of proteins and lipids to distinct membrane domains, ensuring accurate targeting and maintaining cellular polarity. Autophagy, a cellular process involved in the degradation of intracellular components, has emerged as a significant regulator of Golgi homeostasis and function. Under stress or starvation conditions, the Golgi apparatus undergoes dynamic reorganization and fragmentation, allowing its components to be selectively targeted for degradation through autophagy. This process, known as Golgi autophagy or Golgi fragmentation, ensures the elimination of damaged or surplus Golgi membranes and proteins. Conversely, the Golgi apparatus also plays a role in regulating autophagy through the Golgi resident protein ATG9, which is essential for the formation and expansion of autophagosomes. Golgi dysfunction, resulting from genetic mutations, environmental factors, or disease conditions, can have profound effects on cellular signaling pathways. The Golgi apparatus acts as a crucial signaling hub by processing and modifying various signaling molecules, including growth factors, receptors, and enzymes. Golgi localized enzymes, such as glycosyltransferases and sulfotransferases, modify these signaling molecules, altering their activity, stability, and cellular localization. Disruption of Golgi function can impair the processing of signaling molecules, leading to aberrant signaling cascades and cellular responses. These dysregulated signaling pathways can contribute to the development of various diseases, including cancer, neurodegenerative disorders, and immune disorders. After cell division, the Golgi apparatus goes through fragmentation and dispersal. However, it reassembles by organizing fragmented Golgi membranes and recycling Golgi components. Proteins like GRASP and Copy-coated vesicles contribute to the fusion and stacking of Golgi fragments. Understanding this process is important for cell biology research and potential medical treatments. Golgi fragmentation, characterized by the dispersal or collapse of Golgi membranes, has been observed in several neurodegenerative diseases, including Alzheimer's disease, Parkinson's disease, and Huntington's disease. Accumulating evidence suggests that Golgi fragmentation might contribute to the pathological processes underlying these neurodegenerative diseases. Disrupted Golgi function can lead to impaired protein trafficking, altered secretion, and defective post-translational modifications, leading to the misprocessing and accumulation of disease-associated proteins, such as amyloid beta and alpha-synuclein. Understanding the exact mechanisms involved in Golgi fragmentation and its contribution to neurodegenerative diseases is an active area of research, holding promise for developing therapeutic interventions targeting Golgi dysfunction. In addition to complex protein trafficking and post-translational modifications, ion channels localized to the Golgi apparatus also play critical roles in organelle function and cellular homeostasis. These Golgi localized ion channels regulate the ionic composition of the Golgi lumen, pH balance, and maintenance of osmotic balance. For example, calcium ion channels act as molecular switches, modulating protein trafficking, enzymatic activity, and signal transduction processes within the Golgi. Disruption of Golgi localized ion channels can lead to aberrant ion balance, impair the Golgi's ability to process cargo, and compromise cellular functions associated with the Golgi apparatus. In conclusion, the Golgi apparatus is a complex organelle involved in cellular processes like post-translational modifications, protein trafficking, and membrane dynamics. This lecture covered topics like Golgi protein glycosylation, non-canonical secretion, and Golgi's impact on cell signaling and neurodegenerative diseases. Understanding Golgi biology is important for understanding cellular processes and potential therapeutic targets.